Good evening, everybody. Welcome to Science Cafe. We're going to go ahead and get started here. My name is Holly Norris, and I work with Charlie and Jenny King of the UT Southwestern Public Affairs team to produce Science Cafe. On behalf of my colleagues, including our faculty guest speaker this evening, thank you for participating. Science Cafes are online conversations where speakers take you on deep dives into the science of healthcare. As one of the nation's premier academic medical centers, UT Southwestern brings together research, health education, and patient care into one institution. This evening, we're discussing the science of Gulf War illness and Dr. Haley's findings from his 28 years of multidisciplinary research. Before I introduce Dr. Haley, I have a few items to share. We are recording this program and live streaming it on our official UT Southwestern Twitter page. Please mute your microphones to help with audio clarity for all. We encourage you to leave your video on so that we can see each other, especially during the Q&A portion of our program. And finally, just as a reminder, while we cannot answer personal medical questions or policy questions, we welcome your general questions about Gulf War illness. Please post those in the chat. And now I am pleased to introduce our faculty presenter, Dr. Robert Haley. Dr. Haley is a professor of internal medicine and infectious diseases and geographic medicine here at UT Southwestern Medical Center. He also holds the U.S. Armed Forces Veterans Distinguished Chair for Medical Research, honoring America's Gulf War veterans. Dr. Haley received his Bachelor of Arts degree in Philosophy and Social Sciences from SMU and a medical degree from UT Southwestern Medical School. He completed an internship and residency in internal medicine at Parkland Memorial Hospital. Dr. Haley spent 10 years at the CDC, the U.S. Centers for Disease Control and Prevention, serving as a commissioned officer in the U.S. Public Health Service. He received the U.S. Public Health Service Commendation Medal for his research contributions while at the CDC. In 1983, Dr. Haley founded the Divisional Epidemiology and Preventative Medicine at UT Southwestern. His work accomplishments and impact are more extensive than we can share in this brief introduction. I encourage you to click on the program link Charlie pasted into the chat and read more about Dr. Haley after tonight's program. For 3 decades, scientists have debated the underlying cause of Gulf War illness, a collection of unexplained and chronic symptoms affecting veterans of the Persian Gulf War. Recently, researchers led by Dr. Haley solved the mystery, showing through a detailed genetic study that sarin nerve gas was largely responsible for the syndrome. We look forward to hearing from Dr. Haley about his research on tonight's program. Dr. Haley, welcome to Science Cafe. The virtual podium is yours.
Thank you very much, Holly. Let me share my screen and okay, share. Okay, how's that? Does that fill the screen okay? I assume so, okay. All right, so this evening I want to take you on a whirlwind tour of our we just turned 29 years of study on the Gulf War syndrome, from beginning with field epidemiology all the way to new studies on genomics and in between. It's been a long, interesting road, and what I'm in our 30 minutes or so, I'm going to just give you a very whirlwind tour. Obviously can't go into too much detail, but give you some idea of what's gone on these years. Okay, first of all, the 1991 Gulf War. Here's a map of the Persian Gulf. Here's the Gulf here, Kuwait, Saudi Arabia, and Iraq. And Iraq invaded Kuwait, as you may know, in 1990, late 1990. And we sent 700,000 troops over to the theater here with all their high-tech mechanized equipment. And this is actually a map of the unit locations of every little unit on the 19th and 20th of January, just as this war began. Okay, and here are people in their protective suits, because one of the major concerns they had was the exposure to Saddam Hussein's arsenal of nerve gas, which was considerable. Okay. Then 3 years after the war, I received a phone call one day from Kern Wildenthal, who was the president of Southwestern, who said, listen, I want you to come to my conference room tomorrow at 9 o'clock. Ross Perot wants to talk to us. And, Perot had just run for president 2 years earlier. And I said, oh, good heavens, what does Ross Perot want with us? And he said, he wants to talk to us about the Gulf War syndrome and get our advice. And I said, oh, Kern, I've heard a little bit about that. And I think they say it's psychological stress and people trying to get money, and we don't want to get involved in that. It would be career suicide, I said. And so I showed up, and there was Ross Perot, and he said, I've been taking care of veterans for 20 years since Vietnam, and I've never seen anything like this. A whole bunch of veterans coming by my office who I knew before the war were healthy, strong soldiers, and now they're broken people, look like they just came out of Dachau, was his quote. And what we need is an independent study from the government, because my understanding is the government's going to dismiss this. And what if you genius doctors would do some independent studies, I'll pay for it and I'll run political interference. Well, he put up quite a story there, and having spent 10 years at CDC investigating epidemics, if this was anything, it was an epidemic. And so I, against my better judgment, I signed up, and here I am still working on it 29 years later. Anyway, so that's how it started. Okay, what I learned, I went up to the NIH 3 weeks later, and there turns out it was a consensus conference on what was going on with the Gulf War illness. And they paraded in a day and a half, a whole bunch of witnesses that talked about all the exposures and all the symptoms and what they'd learned and what are the possible causes and of this. And here's what I kind of carried away from that. The main symptoms were these: you have, of the 700,000, you have something like a quarter of them have chronic fatigue now, like chronic fatigue syndrome, cognitive problems, attention, concentration, and memory problems, personality change, constant body pain, like flu the rest of your life, balance disturbances, vertigo attacks, unrefreshing sleep and insomnia, hot flashes and night sweats, erectile dysfunction, and watery diarrhea, constant watery diarrhea now for 30 years with intermittent constipation. Notice most of these are subjective symptoms. They're just telling you what they've got, and there's no objective measurement. So 3 years after the war, there still was no objective measurement. Then they went through this litany of possible causes that had been originally inventoried by the Defense Science Board in 1994. It was a group of scientists working, consulting for the DOD. They inventoried and said the possible causes were organophosphate chemical warfare agents like sarin and cyclosarin, pesticides, pesticides sprayed on uniforms, DEET insect repellents, pyridostigmine, and a whole bunch of other things, including combat stress and post-traumatic stress disorder. Strange in that there was almost no personal combat. It was almost all a tank battle, but really it was mostly an air war. So PTSD was kind of unusual, but that was one of the theories. Anyway, so the problem that I saw immediately was we wanted to know the cause. The cause was unknown, so we wanted to know that, but also the disease was unknown, which is really kind of a dilemma in medicine. Most of the time you know the disease and you're just trying to figure out what the cause is. Well, here, both sides of the equation are unknowns. And so we had to figure this out. Well, fortunately, I'd spent 10 years at CDC where this is the rule. CDC deals with new diseases all the time. And so I've done that. So here were the 6 questions that I posed early on of what was going to be our job. First of all, how do you define the Gulf War syndrome? We had to come up with a research case definition. What's the disease process underlying this? Was it psychological, like most people were thinking, or was it organic? What caused it? Was it deployment stress, or was it a neurotoxic chemical injury? And how do you diagnose it? We need a cost-effective diagnostic test. And then how do you treat it to control the symptoms or even reverse the disease process if possible? And then how do you prevent it the next time? So let's start out with the first one, which we did. So how do you diagnose it? Well, I learned at CDC in my 10 years there that the first step in investigating a new disease is to establish a case definition. That is a written, specific definition of what you're going to accept as the definition of disease. And if you can't, and at that time, the entire research community working on this said, well, you can't have a case definition because this isn't a disease. Well, it's kind of circular, you see. Well, as one of my mentors at CDC said, well, if you can't, then establish a case definition, because until you've established a case definition, you haven't begun the research. So 3 years after the war, nothing really productive had been done. Well, this is a roadmap of what we've done over the last 29 years. Okay, and here was the first phase we then started initially based on that case definition to establish it and then to see if we could start answering the questions. Let me just go through this quickly. Then later, I'm going to talk about the next phase where we developed the methods for a national survey to see if we could replicate our definition in the whole military population. And we surveyed 8,000 randomly selected veterans, collected blood from 2,000 of them, all the cases and a random sample of the controls. And we developed a whole bunch of new neuroimaging tests and other kind of tests to try to show what was being affected and prove what the disease was. And then we assembled two groups, a group that we studied originally, and then a sample of national from all over the country. So it would be representative of the country. Okay, and then we did some animal studies and so forth, which I won't talk about today. And then finally, we're now doing some gene expression genomic work. Okay, so that's where we're going. Okay, so for the fetal epidemiology, I developed a questionnaire booklet of the symptoms and exposures. Okay, here's the exposure booklet and the symptom booklet to inventory or ask each veteran, did they have the symptoms that had been described in that NIH conference and in our other initial studies, and also the exposures. Did any of them have those exposures that were thought to possibly be the cause? And we then signed up a battalion, a reserve battalion, that when they came back, they were no longer in the military, so we could get to them without permission from the Defense Department, which we wouldn't have gotten at the time. So we got this battalion, and they agreed to work with us. We recruited 250 of them. We met them out in the southeastern states. They filled out the survey booklets, and we then we carried out a statistical procedure called factor analysis or principal components analysis, which looks for patterns in the symptom data. And sure enough, we found 3 what looked like syndromes. So this became our case definition. And there were 3 of these 3 different variants, we think, of the Gulf War syndrome. So our hypothesis then was there are 3 manifestations of the Gulf War syndrome that were very much related to each other on a background of veterans who developed other illnesses that they would have gotten anyway. So not Gulf War related, but they thought they were. And then the background of most veterans still well. Some of the veterans got sick while they were still in the deployment, but others after they came home, and then after a few months, very few got sick after that. Here's the amount that were unemployed at the time we surveyed them. And the first syndrome 1 and syndrome 3 groups were a little less employed, but the syndrome 2 were really sick people. We then brought in some of the sick ones and some of the well ones and did a comparative study with a lot of neurological tests and neurophysiological tests, objective markers, and found that sure enough, the controls were different from the cases. The cases a little bit more impaired on all these tests, suggesting that it really is a neurological problem, not a psychological one. So we then did an analysis of the exposures, and we found for syndrome 2, the most severe, the hearing nerve gas alarms going off, being exposed to low-level nerve gas that didn't kill anybody. But that exposure was the strongest predictor of having this illness, and are many side effects from the anti-nerve gas tablets that you would predict the symptoms would be increased if they'd already been exposed to low-level nerve agent. So those really suggested from an analytical viewpoint that nerve gas was really the cause of the second syndrome and probably also the third one. So nerve gas emerged then as our #1 theory. So we published 3 papers. These are the 3 title pages these three papers collected together in Journal of the American Medical Association in January 97. And this really was like a nuclear explosion because the government had decided this was psychological and they didn't have to do anything about it. And suddenly here's these upstarts coming up working for Ross Perot saying that it's actually a real illness, which they defined here. It's got neurological components to it, neurological deficits related to it, and it's related to being exposed to nerve gas. So this was very shocking and actually brought out a very vitriolic response that lasted a number of years. Okay, so the second question, what's the disease process? What is this disease? So here we then had to go back to the workshop and bring our cases and controls back in, do some more testing on them. And then we developed the methods for a big national survey so we could recapitulate the questionnaires in a random sample of all Gulf War veterans to see what was really going on, if this was true of all or just of that special unit. We collected blood from a quarter of them, all the cases in a random sample of the controls. And then we developed a whole bunch of new neurological and neural imaging tests with brain imaging to see if we could actually visualize the illness. Just to give you a smattering of some of the findings, here is when we do a 24-hour electrocardiogram, an EKG. We put the leads on and record the heartbeat for 24 hours. Here's early morning, 7 o'clock in the morning, and then up to 7 o'clock the next morning. And we measure this with a statistical technique that measures the amount of variability in the rate of the heart rate. The heart rate varies as you take a breathing in and out, and that is regulated by a brain process that is the parasympathetic nervous system, part of the automatic nervous system of the brain that regulates much of your body's function, unconscious functions. This regulates your digestion, your elimination, your body temperature, and a whole bunch of automatic functions. And what we found is at night, when they were asleep at night, the well veterans had a big increase in heart rate variability, which is normal. And this indicates the restorative aspect of sleep when the parasympathetic nervous system kicks in and kind of cleans your brain up and makes you get ready to wake up the next morning. Whereas look, the sick veterans were flat. That didn't happen. So there was something wrong with their parasympathetic nervous system. And this, we think, explains why a lot of them have unrefreshing sleep. They may sleep all night, but they wake up hammered like they didn't sleep. And we think it's because they didn't get that restorative function. Also, this may explain the chronic diarrhea because this same function regulates your intestinal tract and may be a neurological cause of diarrhea. Years later, when we did our national survey, we did the same thing on a bunch of cases and controls. And sure enough, here are the controls, and they're normal increase at night. And here are the cases, and look, pretty flat. So this was true of all Gulf War veterans who have Gulf War illness compared to the ones who don't have it. Here's a really interesting study where we administered a medicine that stimulates parts of the brain that we think are probably damaged or abnormally functioning. And we do a brain scan before and after that. And then compare the sick veterans and the well veterans and see these little parts of the deep brain. These are parts that were functioning differently in the sick and well people when they were stimulated. So basically the sick veterans did not stimulate normally in the areas that normally would be stimulated by this medication. So suggesting even the chemical reason for this. And actually, here's a graph produced by some of my SMU statisticians who did some very inventive work on this. Here's the syndrome 1, the syndrome 2, and the syndrome 3 predicted by those response to that medication. And you see the syndrome 1 and syndrome 2 are opposite sides of the control group in the middle, but it distinguishes the three groups from the controls. But notice if we hadn't separated these into three groups and just averaged them all together, they'd all be sitting on top of the control group and you wouldn't see anything. So this shows that the initial factor analysis that we did was really important. We then got intercession by Kay Bailey Hutchison, who had been following along and liked what we were doing and was our senator. So she got a bill passed through Congress that funded us for a big increment in funding, and we were able to actually buy our own brain scanner. Here it is being loaded off the trailer, and we put up as the first 3T magnet west of the Mississippi River, 3 Tesla. It was a very powerful magnet at the time. We invited a bunch of universities to come in and develop their own brain imaging techniques if they felt they could come up with a functional MRI or some other brain scanning technique that would explain each of the symptoms of Gulf War illness, what part of the brain is malfunctioning. And so we got some good takers from those universities. And just a few highlights. This is a chemical analysis of a small part of the brain, which we had found looked to be damaged or malfunctioning. And what you get from this brain scan is a chemical profile. Okay, and this is a profile of a normal variant, a control of normal veteran. And see this big spike of the chemical NAA? Here's one of our Gulf War veterans who was really sick, and see the little spike is kind of puny, suggesting that these brain cells are not healthy. So we had some now chemical evidence that this was a disturbance in the brain. Here's one by a group at the University of Texas at Dallas that are very inventive functional MRI. They put veterans in the scanners, the case veterans, the sick veterans, and the well veterans, and compared them. And each veteran would go in the scanner, and while they were being scanned, we would ask them, or a voice would come on and ask them to memorize a list of common items. They had to memorize the list, and then later, while the scanner was still going, they would have to recite what that list contained. So it tested their encoding of memories and their retrieval of memories. And sure enough, the cases and the controls differed, because remember, memory was one of the main symptoms, difficulty remembering things. And sure enough, the sick veterans, the well veterans, lit up this little area in the prefrontal cortex, which normally processes memories like that. And whereas the sick veterans didn't. So this area here of abnormality, this is the front view and this is the side view. So this shows, this is the area where the cases and the controls differed. Okay, so indicating that the normal process of trying to encode and retrieve memories was not working well in the Gulf War veterans. So they're not making up this complaint of memory problems. Their brain isn't doing what it's supposed to do when they're trying to remember, encode and remember something. Similarly, we did another functional MRI test like that, putting people in the scanner and having them try to come up with words, because word finding was a big complaint of sick Gulf War veterans. I just can't think of the words. I can't think of names anymore that I used to know very well. And so we would, in the scanner, we would say, tell me all the ocean animals you can think of, whale, dolphin, crab, et cetera, and see how many they could name in a few seconds. And then give them a word and say, how many words can you find that rhyme with that? So they have to come up with words, see? And sure enough, the cases and controls differed in the normal area that processes finding of words in these deep gray matter brain structures. These are different slices of the brain, and it shows areas that are where they differ. We then did some high resolution electroencephalography. This is the EEG capturing all these different areas of the brain, and here are all the brain wave tracings. And our Jeff Spence who was on our staff, who'd come from SMU statistics department, has analyzed this and found different patterns in the sick veterans and the well veterans. So we had a really good idea now that this was a real organic brain disease, not a psychological thing. We kind of knew the areas of the brain that were malfunctioning here, at least some of them. So now the big question that I really want to deal with today in the next few minutes is, what caused the disease? That was the big question at the beginning. Was it deployment stress, or was it a neurotoxic chemicals? Okay, well, the first question was, were the US soldiers actually exposed to low-level nerve gas? This was very controversial. If you ask a sick veteran as they were coming home, they say, oh, absolutely. man, it was all over the place. Our alarms were going off different times. They were going off, and we never got really sick and nobody died, but those alarms were going off. And so we think that was probably the cause. So that's what everybody thought. Well, right after the war, this fellow here, Jim Toot, was an intelligence expert who was hired by Senator Don Regal of Michigan, who headed the Senate Banking Committee. And he was doing the first investigation of Gulf War illness in 1994, about the same time we got started. He hired this guy to do the investigation, and they were issuing their report in 94 when I was up there for that NIH conference. And I sat and listened to that. And he basically said this. Here's the map. And he said, on the third night of the air war, so there was a 5-week air war and then a 5-day ground war. So from the middle of January, the 5-week air war, on the third night, we bombed the Iraqi chemical weapons, the sarin production and storage facilities up near Baghdad, okay, and blew it all up. And then the next morning, about 10 o'clock, 10,000 nerve gas alarms started going off, and various experts from other countries who had very sophisticated chemistry equipment and trailers were actually detecting sarin nerve gas in the air. So there's no doubt there was sarin there. And it happened on the morning after the third night of the air war, okay, when they bombed the chemical plants. And it continued on for at least a week or so and so this was his theory that was showed that it actually happened. Well, 10 years later, he hadn't published that. Nobody believed it. And so I said, Jim, we had become good friends by then. And I said, you've got to publish that. And he said, well, I don't write scientific papers. And I said, well, I can help you. So we sat down and collaborated, and we came up with a lot of evidence, explained how the nerve gas got down there. It turns out the wind direction was right and other conditions were right. But then I said, we need a smoking gun. So he had the idea to go over to NOAA, the National Oceanic and Atmospheric Administration, and look for weather satellites that were going over that night that might have detected this fallout cloud. And sure enough, here it is before midnight. And then after midnight, you see a little bitty wisp occurring right here. And then it gets bigger and bigger every 2 hours or 4 hours or so. And by the next day, it was this big cloud, big fallout cloud that was reaching all the way down where this is where our troops were positioned. And here it is reaching down almost to our positions there. And that's about when the nerve gas alarm started going off. And here's an enlargement of the last picture that he got. And here is the debris cloud. See, the big yellow clouds here are just a normal cloud bank. There was a weather front right here. But here, this dirtier yellow, lighter yellow cloud is the fallout cloud. It was rising from here and then gradually moving down, enlarging down to here until it got to here. Here's where our troops were stationed, all down in here. As you can see, it eventually enveloped the whole area here. And so that's a smoking gun actually showing the fallout cloud from the bombing. So this really changed we published this in a very good epidemiology journal. And this basically shut down all the discussion of whether this actually happened or not, whether they were exposed. Basically, our entire 700,000 troops were exposed. Okay, so then we start the next phase. We actually, this was then funded by the VA with after legislation by K. Bailey Hutchison that appropriated additional money for this through the VA. So they funded this to actually carry out the national U.S. military health survey that we had designed along with Research Triangle Institute, and collect blood samples from 2,000, all the sick veterans and a random sample of the well ones, and then pick some subsamples that we could do lots of bring in to do some very extensive brain imaging and other kinds of studies here in our hospital. Well, the question is there a way to prove that sarin nerve gas was really the cause? And that's a big order, because you may have heard over and over the old expression, in an observational, when, a clinical trial where you randomize people to treatment or placebo, that's kind of the gold standard for proving whether a drug really causes the disease to get better or causality. But if you have to do an observational study where you can't randomize, the people say, well, what you get out of there is just a correlation, and correlation does not prove causation. You may have heard that. Well, that's generally true. However, when you're dealing with genes, that changes the equation, because think of this, whether or not you have the strong form or the weak form of a given gene is determined randomly at birth. God assigns you to the strong form or the weak form of every gene, and it's just random at birth, at least random over a population. And so if you find a gene that's associated with your certain cause, then that suggests that actually is a causal inference and is very convincing. Well, very soon after we saw that, our first study showing that sarin nerve gas looked like it was the cause, but we couldn't prove it yet. I did a quick computer search of the medical literature with Medline through our excellent library here at Southwestern. And I just simply crossed sarin nerve gas genes. It was no big deal. And I got a whole bunch of papers on this gene that protects you from nerve gas. And it was the paraoxonase 1 gene, abbreviated the PON1 gene. And this has a little difference in it called a polymorphism, where you're either born with the Q form or the R form of the gene. And the Q form is very strong. It makes an enzyme that destroys nerve gas, whereas the R form is the weak form. The enzyme it produces is not very good against nerve gas. So this was a perfect finding. And most of those papers were written by this guy, Bert Ledoux at the University of Michigan. He was the grand old man of paraoxonase. So I just called him up and said, Dr. Ledoux, would you like to collaborate? I'm doing this study on Gulf War syndrome. I'd like to send you the blood samples and have you measure the gene, whether the strong form or the weak form of the genotype, and could you measure the enzymes, the Q enzyme or the R enzyme, and tell us who's really susceptible and who's at risk, and who's not susceptible. And we'll see if that's related to Gulf War illness. And our theory was that the sick veterans would be ones born with the R form, the weak form of the gene. And so when the low-level nerve gas came over, it didn't kill anybody, but it got through their blood to them and made them sick. Whereas if they had the Q form, this would destroy it. It wouldn't get to their brain and they would be protected. Okay, that was the hypothesis. So anyway, I sent him the stuff. He did it, and here is the level of the Q isoenzyme. So the higher this is, the more you're protected. Okay, and here's the control group, the ones who are well, the syndrome 1, 2, and 3, the 3 with Gulf War syndromes. Okay, and you see here, most of the well veterans had very high levels of this, whereas the ones with who are sick had lower levels, and this was highly statistically significant. Half these were deployed to the Gulf and the other half were not deployed. And it turns out these 4 were not deployed. So what do you think would have happened if they'd been deployed? I think they'd have been over here somewhere. Anyway, but it was enough to show that there really was an association. Here's the averages in each group. And you can see the controls are much higher than the syndrome, and syndrome 2 is the lowest. We were the sickest guys. So this looks like a real association between a protective gene. So now here is the association of the number of nerve gas alarms that each veteran saw. We asked them, one of the questions was, how many of these nerve gas alarms did you hear? How many times did your alarm go off? And it went anywhere from 0 to about 30 or 40. And so from 0, 1 alarm, or 2 to 9 or 10, here's the risk, the relative risk of having Gulf War illness. Compared to those with heard no alarms, here's the ones that just heard 1 alarm, 2 to 9 alarms, and 10 or more alarms. You see, just went right up like that. And here's the genotype. If you had the protective genotype, the QQ, you had a low risk of Gulf War illness. If you had the R form, you had the highest. And if you had a mix, you were halfway in between, partway in between. So, I mean, it worked out perfectly. Now, the question is, does this interact? Is this gene, is what it's doing, it's modifying whether the nerve gas alarms can make you sick or not? Okay, that's the question. And here is the analysis. And I know this is, in the short time, it's going to be hard for you to conceive this, but let me just go through it and see if you get it. So here are the sick veterans and the well veterans. Okay, the cases and controls. Here is, did they hear nerve gas alarms, yes or no? And here is the lowest quartile of the Q activity, the ones most at risk. Here are the ones with the highest amount of the Q isoenzyme, the most protected. And here's the mid-high and the mid-low. So in terms of the most protected and the least protected. This then is the relative risk of getting Gulf War illness. So here, if you had the highest protection by the gene and the enzyme, okay, you had hearing nerve gas alarms caused you a 3-fold higher risk of Gulf War illness than if you didn't hear nerve gas alarms. Whereas if you had the lowest protection from the gene, the ineffective form of the gene, you had an 8-fold increase in. So you see, the gene then modifies the ability of hearing nerve gas alarms or low-level nerve gas to make you sick, to give you Gulf War syndrome. And this is what's called a gene-environment interaction. And that in medical science is gold. Here's just a graphical representation. Here's the highest amount of the Q isoenzyme, the highest protection and the lowest protection and intermediate. And here's the relative risk, low, mid-low, and then way up here for the lowest level of genetic protection. Hearing nerve gas alarms had the most effect on the most veterans up there. Okay, so that's a gene-environment interaction. So that was our main finding in our paper. And there were 2 remaining objections to that finding. And that is the thing called recall bias. And this had been brought up over and over for years and years by the critics and by the skeptics of this, who didn't believe there was such a thing as Gulf War illness. And they say, oh, there's recall bias. Recall bias is an epidemiological phenomenon that we know about when you do epidemiologic studies, when you're asking about an environmental risk factor. Were you exposed to something? And we know that when people are already, usually we are doing these studies when we have some sick people and some well people, and people already know they're sick. Well, the sick people, if you ask them, were they exposed, they're sick and they're wondering, why was I sick? And they're constantly thinking about it, you see. And so they're very likely to remember those exposures, whereas the well people are not thinking about it. And so you ask them and they don't even remember what was going on back then because they've not been thinking about it. And so they're likely to undercall it. See, many of them will say, no, I didn't hear any alarms going off, when in fact they did. And so that misclassification, that can cause a false positive association. And that's one of the reasons that in that kind of a study, correlation does not prove causation. Okay, it can be due to recall bias. Well, the question is, yes, that's true if we were just looking at the association of nerve gas exposure with the Gulf War, but we're looking at the gene-environment interaction. And so what's that effect? So one of the things in our paper, we did an analysis based on some relatively new theory in epidemiology that is able to disentangle that, and we were able to prove that recall bias could not have biased and explained away our finding. And finally, there's the old bugaboo of the unmeasured confounding variable. That is, yeah, we controlled for age and sex and a bunch of confounding variables that we could measure, but was there another confounding variable out there that really explains the findings that we didn't measure? Well, it turns out in relatively new literature, there is a way of exploring that, and we did, and found out that the strength of the gene-environment interaction was so strong that no unmeasured confounder could be strong enough to explain it away. So we defeated the two main arguments that would have invalidated our findings. So this then gave us a conclusion that we now have evidence from a genetic study, gene-environment interaction, that proves that sarin was a major cause and maybe the major cause of Gulf War illness. Finally, answering the main question that we started out with. And here was the paper, evaluation of a gene-environment interaction of the PON1 and low-level nerve agent exposure with Gulf War illness, a prevalence case control study drawn from the U.S. Military Health Survey's national population sample. Furthermore, see, this was done in a national population sample of veterans, not in some little grab bag group, but in a random sample of Gulf War veterans. Okay, and so that was published in May. It was published along with an invited editorial by Mark Weisskopf, who is the head of environmental health and epidemiology at Harvard School of Public Health, one of the most outstanding researchers of environmental illness, who's written a couple of papers on Gulf War illness, by the way, and Kim Sullivan, who is all at the same position at Boston University School of Public Health and has done a lot of work on Gulf War illness. So these are some of the top people in the world on this subject. Here is the conclusion of their perspective, commenting on our paper. In summary, the author's exploration of a gene-environment interaction between presumed nerve agent exposure and the PON1 gene offers some strong arguments that there is a true causal effect at work. It also suggests, at least in part, why some soldiers who were presumably exposed to toxins like nerve agents suffer from Gulf War illness and some do not. So basically, this was an editorial that gave as strong an endorsement of our findings as you can find in a journal. So I was recently up at a meeting at the VA central office, and one of the top directors of the VA heard my talk where I presented this information, and his comment was, "This paper has changed everything." Now, we're now on to another phase of the research, trying to get at how to develop a diagnostic test and to figure out then from all these findings, is there a way to treat this? And just briefly, here's a heat map from a typical gene expression study looking at the difference in gene functions across the genome. Here's a bunch of genes over here, and here are the soldiers. And you see here are the Gulf War veterans with these two different patterns, and here are the controls with nothing like that. And so, and what we're finding, what these genes are that really distinguish the cases from the controls, are things called cytokines and chemokines. Basically, they're inflammatory proteins. They're not evidence of brain cell damage. They're evidence of inflammation. So what we think is the sarin gas got up in the brain and sensitized some of the immune cells of the brain that then are constantly setting off inflammatory products, what are called cytokines and chemokines, which are the things that make you feel like you've got a cold when you get sick. With a cold or flu, your body starts making these things, and that's what makes you have fever and shiver and have aches and all that. But this is happening up in the brain, and this is very positive and optimistic, because this may not be brain damage that can't be fixed. This may be inflammation, and we now have many ways of turning off inflammation that we don't want. I mean, think of treating arthritis with these new drugs you see on television all the time. These are very effective at turning off inflammation. So if this holds true, and we're still trying to enlarge these studies and confirm them, if that's true, then this sets us on a way for a possible treatment. And we're now putting in a grant for a thing to develop a diagnostic test, which we hopefully will be funded by the Defense Department, and we're off and running. Let me just give some credit to people who've been so key. Kern Wildenthal, was president of UT Southwestern back then, who got me involved and was very supportive all the way through. Ross Perot and the foundation, Senator Kay Bailey Hutchison, longtime colleagues in my laboratory, Jerry Kramer and John Talbert. I must say, from our initial studies, very first studies in that field work, Tom Kurt and Jim Hom were two people who were instrumental in helping us do that first stage, get us going in the right direction. And then over 100 faculty research colleagues from UT Southwestern mainly, but some other universities, Research Triangle Institute in North Carolina, for collaboration in various phases of the project. Funding from the U.S. Army Medical Research and Materiel Command that's funded us on and off and actually very steadily for the last 10 years through their peer-reviewed program, and the U.S. Department of Veterans Affairs. And let me say, the rich intellectual environment of the UT Southwestern campus, where for any new technique you want to learn, there is a national expert here willing to teach you or collaborate. This is a truly unique medical research campus that just makes this kind of research possible, where it probably wouldn't be possible at most other places. Okay, that's pretty much it. I think we have time for questions.
We do. Thank you so much, Dr. Haley, for your presentation. We will now begin the Q&A portion of this session, starting with questions submitted ahead of time before we address questions in the chat. But first, Dr. Haley, I wanted to let you know that one of our registrants wanted us to share with you their thanks and appreciation for your commitment to this important project, and we join them with our thanks.
Well, good. Well, let me tell you, it's been a great honor to serve all the veterans who in turn have served our country. And I'll tell you, you've never seen a more appreciative group than the veterans who have this disease. I'll tell you, it's been very rewarding.
Yes, thank you. Thank you for your presentation this evening. So our first question regards if there is research or literature showing any link between Gulf War illness and multiple myeloma.
Good question. There are a number of diseases that one would wonder about, and so far there is no association with multiple myeloma. Interestingly, we did some studies early that showed an association with amyotrophic lateral sclerosis, or Lou Gehrig's disease, which is a horrible degenerative disease of the nervous system that's fatal usually in about 2 years. And there are a bunch of, we recognized this early on from our surveys when we found a bunch of very young Gulf War veterans in their 40s who were getting ALS, and that's usually a disease in the 50s or 60s, 70s, and so forth. And we found some uncharacteristically young ones and then did a national study of it, and sure enough found that it was strongly associated with having served in the Gulf and having Gulf War syndrome earlier. And then the VA took on a project to try to disprove us and ended up finding the same thing, and that resulted in the VA service connecting all cases of ALS in veterans of any service. They're now service connected and get care in the VA when that was not happening before.
Okay, thank you. Our second pre-submitted question is Gulf War illness also seen in people living in Kuwait during the war, or in those who went back once the war was over?
Oh, good questions. There's no evidence, period. Basically, there's one study, but basically the people who were studying it really didn't have entree to what they needed to actually study it. And remember, those countries are not free democratic societies like ours, where you have a Ross Perot and a Kay Bailey Hutchison who will make sure studies get done when they need to be. And those are basically dictatorships, Kuwait and Saudi Arabia and Iraq. And so we really wouldn't know if there were or not. I suspect there are probably a lot of them, but we just don't know. We'll probably never know.
Thank you. Our next question comes from Thomas Kurt, and It's actually not a question, but he wants you to describe how group 2 were exposed to sublethal concentrations of sarin, plus were taking PB and presumed antidote on
Right.
the orders of their company commanders.
Yes, Tom Kurt. I mentioned earlier, Tom Kurt was one of our collaborators. He's a toxicologist of some national renown who I called upon right at the very beginning to collaborate with us, and he did very effectively. It helped us work out a lot of the hypotheses that turned out to be right later on. And so it's very interesting. There had been some scientific experiments for a number of years before the Gulf War to try to come up with antidotes for nerve gas poisoning. And one of them was to give a medicine called pyridostigmine bromide, or its common name is Mestinon. That's a drug we give to people with myasthenia gravis. They thought it was supposed to, if you gave it before the exposure to sarin or to nerve gas, it would protect the receptors that nerve gas binds to. And then if you could get them to medical care pretty quickly, then the drugs that you give to treat it would work better, and it would actually reduce the mortality rate from nerve gas attacks by 50%. Turned out that was, and so as a result, the military gave low doses of that medicine to about half of our troops in there, because when the alarm started going off, they thought maybe they were in for nerve gas attacks. And so they started giving them these pills to take, and they were getting a lot of side effects from them. And it turns out, while they were actually right there about that same month, a paper was published in just a regular chemistry journal showing that if you give the pyridostigmine, the antidote, after the nerve gas exposure, it actually makes the nerve gas exposure worse. But they didn't know that beforehand. That study had not been done, but it was being done just as we invaded Kuwait, and it was published almost the same time the invasion was occurring, but it was way too late for anybody to read it and put that into practice. So there's some evidence, at least theoretical evidence, that the pyridostigmine anti-nerve gas pills might have made the illness worse. The epidemiologically, the evidence doesn't really strongly support that, but it's kind of equivocal. So it could be, it might be, but it might not be. But anyway, it was a very good point.
Thank you. Our next question comes from Amila. She wants to know, how long was sarin in the air? And this is a two-part question. How long was sarin in the air, and how long do you have to be exposed, or did you have to be exposed to sarin for you to suffer?
Oh, very good question. Let's see. I didn't have the slide in there for that, but I have a slide that in the paper I published with Jim Toot, the one showing that nerve gas actually was, the exposure actually did occur, from that cloud of fallout. We have a figure in that paper taken from EPA, the EPA regulations on sarin exposure in laboratories. So if laboratories are going to do experiments on sarin, they've got to be sure that any exposure would be below a safe level. Okay, and so they publish what are the safe levels, and it turns out that the level that can be detected by the nerve gas alarms to go off, it's a fairly high level. So when the nerve gas alarms go off, people have already been exposed to a low level of nerve gas. And then after the alarms stop going off and it starts dissipating, for a while then, they're still being exposed to lower levels, which now we know are dangerous. So the nerve gas alarms were set too high, but again, nobody knew that at the time. And so basically when that nerve gas cloud came over, the soldiers were being exposed pretty much for hours at a time, but it would kind of come and go. And we think, now this is not proven, but we think one reason that it was the exposure was intermittent was one, the alarms would go off for a while, and then they'd stop going off, and then they'd go off again. And that went on for about a week, that we know. But what we think's happened is the nerve gas was up way high up in the air. And then when explosions occurred, there were Scud missiles going off and all kinds of things. When something would disturb the atmosphere, it would mix the layers of air, and some more sarin would come back down to the surface, and then it would float back up, and then something else would happen. And so we think something like that was happening. But again, that's all speculation. But there was intermittent exposure on and off for about a week.
Thank you. Now I'm going to go back to one of our pre-submitted questions. The question is, what technological advances in understanding genetics helped lead you and your colleagues to the research findings about sarin gas?
Wow, that's a really great question. One thing it shows is the great power of the advances in medical research technology that over time allow greater and greater insight into disease. Had we tried to do all these studies that have more recently occurred, had we tried to do those 25 years ago or 29 years ago, we wouldn't have been able to discover what we discovered, because those techniques didn't exist back then. There was some ability to identify the genes, and actually the PON gene was known back then, was well known back then. But the epidemiologic theory and the statistical methods for showing that this was a causal association had not been conceived yet. And so that happened about 10 or 12 years ago. And now the techniques that we're starting to employ right now to measure gene function, how well are all the genes in the genome functioning, and then to measure all the proteins in the blood, so we can find a combination of proteins that'll become a diagnostic kit, a blood test kit. Those techniques are just emerging in the last several years, and basically I'm still finding new statistical techniques to analyze these very complex, very large. This is large data. You've heard of large data. Well, this is very large. This is 150,000 genes for each person, and we're trying to find a needle in that big haystack. And the statistical techniques are not well worked out yet, and are evolving. And I've just recently discovered a new paper that gives us a much better way to do that. So these things are still unfolding, and maybe next month I'll be able to do something even better.
Thank you.
Let me say one other thing. As I mentioned earlier, being in a place like UT Southwestern is just cannot be underestimated, because we have a core laboratory that can measure 150,000 genes, the RNAs that show how they're functioning, and they do that for anybody on the campus who has funding for a study to measure gene expression. We just send the blood samples over to this core lab, and they do all the measurements, send it back to us, and we have to figure out the analysis. And similarly, we have a proteomics core that can measure 7,000 proteins in a blood sample, and then return us that data on any number of veterans, and then we do the analysis. So without those facilities, we could never hope to do these kinds of studies. And back when we set up our brain imaging facility, and Kay Bailey Hutchison got the funding for us to buy our own scanner, we were able to get a state-of-the-art scanner, and actually had space to create a new brain imaging center. And the university then could actually carry that off, could do that. We had the construction people, the contracting people, everybody in place who could actually make that happen. And so only in a place like this could this research be done.
Thank you, Dr. Haley, for sharing your results of your 28 years of research into Gulf War illness. Your work is absolutely remarkable. I'll turn the podium back over to Holly to close us out.
Thanks so much, Charlie. And I concur with your thanks to Dr. Haley for joining us tonight to discuss his complex research and findings. To our guests, we're glad you joined us for another episode of Science Cafe, and we appreciate your questions for Dr. Haley. I would like to thank Charlie for helping facilitate tonight's program. Before we adjourn, we invite you to join us again in 2 weeks on November 17th for our next Science Cafe. The topic is regarding non-surgical treatments for sports injuries and other musculoskeletal conditions. Click on the link Charlie pasted into the chat to register for the program. As we conclude for our evening together, we wish you all good health and wellness and a good night. Thanks again for joining us. We're adjourned.